Hi again, everyone. Thank you for joining today's webinar hosted by Ramona, where we will be discussing how to automate high-throughput organoid and zebrafish screening. My name is Natalie Alvarez, and I am product lead at Ramona. I'm excited to be your moderator for today's webinar. Our goal is to make this webinar both educational and enjoyable. At Ramona, we like to do things a bit differently, so today will be no exception. We're going to have some polls sprinkled throughout the session, and we will offer prizes to the most engaged participants, including those who answer all of our poll questions and also ask thoughtful questions in our Q&A. So for our first poll, hopefully this is an easy one. What prize would you want to win today? We'll give everyone a moment to answer here. Thank you so much for your input, and stay tuned for some pop quizzes later in the meeting. This webinar will highlight how our technology can transform your manual workflows into automated assays. And we're excited to share 2 case studies on this from leading organoid and zebrafish researchers. Before I introduce our amazing speakers, I want to quickly introduce our technology at Ramona. Our goal at Ramona is to help you go from images to insights in a matter of seconds, not minutes or hours. And to do this, we've created a paradigm in microscopy. So when we reflect on the history of microscopy over the past 150 plus years, very little has actually changed. Existing optical microscopes remain largely confined to one objective lens for one human to peer through. Automated motion and digital cameras have increased throughput. However, state-of-the-art high-content platforms continue to utilize a single objective that repeatedly steps and scans over multiple fields of view. However, for large samples, that process is very slow. And for live samples, it's tedious and difficult to capture dynamic changes happening in real time across wells of a well plate. Enter the world's first gigapixel microscope. With a call to action from the NIH, our team set out to break that traditional trade-off that has existed between field of view and resolution, with an initial vision of examining freely swimming zebrafish with high enough resolution to pick up neuronal activity. The solution was to invent an imaging system that was fast for large samples and both easy to use and, importantly, non-destructive for precious living samples. So what we've done is essentially leverage the evolution of camera technology as it has reached small sizes for cell phones, and we've densely packed those cameras into an array that then we put into compact benchtop systems. This multi-camera array microscope, or MCAM technology, enables the cameras to work synchronously to capture live biology. The visual on the right demonstrates the array for our Vireo system, which has 24 cameras, and provides a sense of the speed at which it can scan a 96 well plate as compared to a single objective system. Our MCAM technology powers 2 products, the Kestrel and the Vireo imaging systems. The Kestrel was designed to be the most comprehensive system on the market for analyzing behavior, morphology, and fluorescence at video rates in model organisms like zebrafish. The Vireo was designed to be the world's fastest live cell imaging system with single cell resolution. And it's particularly well suited for quality control applications, high throughput drug screening, and real-time cellular dynamics. Now, I am excited to introduce our speakers for today's webinar, both of whom are early adopters of these 2 products. We're excited for them to highlight their use cases and explain how they've used Ramona's imaging and analysis tools to accelerate their research. Our first speaker, Dr. Robyn Tanguay, is a distinguished professor in the Department of Environmental and Molecular Toxicology, the director of the Oregon State University Superfund Research Program, director of the Sinhuber Aquatic Research Laboratory, and director of an Environmental Health Sciences Center, all at Oregon State University. She received her BA in biology from California State University, San Bernardino, her PhD in biochemistry from the University of California, Riverside, and postdoctoral training in developmental toxicology from the University of Wisconsin-Madison. She serves on a number of academic, commercial, and federal advisory boards and is on the editorial board for several scientific journals. Over the past several years, she has pioneered the use of zebrafish as a toxicology model and recently developed automated high-throughput instrumentation to accelerate phenotype discovery in zebrafish. One of her primary focus areas is identifying chemicals and mixtures that produce neurotoxicity. Phenotypic anchoring, coupled with the inherent molecular and genetic advantages of zebrafish, is used to define mechanisms by which chemicals, drugs, and nanoparticles interact with and adversely affect vertebrate development and function. Thanks for being here, Robyn. Our second speaker, Dr. Jason Stein, is an associate professor in the Department of Genetics and Neuroscience Center at the University of North Carolina at Chapel Hill. He is principal investigator of the Stein Lab, which explores how variations in the genome change the structure and development of the human brain, and in doing so, create risk for neuropsychiatric disorders. He received a BA from Northwestern University in the Integrated Science Program, and then pursued post-baccalaureate work in the intramural program of the National Institute of Mental Health with Andreas Meyer-Lindenberg. He received his PhD in neuroscience from the University of California, Los Angeles under Paul Thompson, where he worked on discovering the genetic influences on macro-scale human brain structure. He also completed postdoctoral training at the University of California, Los Angeles under Dan Geschwin, where he developed tools to evaluate how neural stem cells model brain development. And he also studied regulatory regions governing human neurogenesis. Without further ado, I'm excited to hand it over to our first speaker, Dr. Robyn Tanguay, to describe how her team is using our Kestrel system to image and analyze zebrafish behavior and movement.
Okay, I hope you can see my screen okay, and really appreciate the opportunity to present kind of our research goals and how we've integrated the Ramona technology to really impact our group's activity. So the title of my talk is really the goal of this part of my lab is scaling in vivo chemical bioactivity screening, and I'll show you why we think this is important. So when you think about, why did we use zebrafish as a research model? We realized that what the field really needs, the field of toxicology in general, and chemical biology, is to have a biological system that would be useful for evaluating chemicals with very different uses or structures or modes of action. So when I made the decision years ago to use the zebrafish model, we realized that early embryonic development is the most dynamic life stage of any organism. And so we know that from practical experience that early life stage, say human development, are more susceptible to chemical insults. And it's really operationally because in the early life stages, that's when most of the genome is present and it's required to accomplish the amazing process of embryonic development. So if you want to kind of put it on its end, that realizes that if you're going to throw a chemical at a system and you have no idea what the target of that chemical would be, the best time to do it would be during embryonic development when the entire system is present and operational. So what we really think about when we wanted to build a workflow is we want to capitalize on this complexity of biology, and we're going to throw chemicals, lots of chemicals individually in mixtures across concentrations to this system and dare the animal to develop normally. And so when we identify an adverse outcome, that tells us that we should take a closer look at that chemical. So when we thought about doing this, we really wanted to make sure that we can do it all in a single cell, in a single well. So we want to get to the point where we can extract as much information as we possibly can from sacrificing this vertebrate. So we want to put an individual animal in a well, and we want to extract across concentration whether or not there's early effects on behavior. So we have instruments, and the Kestrel is actually going to be useful for this as well. We capture an early behavior at 24 hours early in development. And then we let the animal develop for another 4 days when now you have this really complex organism where most of the organ systems are developed. And now we said, what can we extract from each one of these animals in real time? And so we have a number of behavioral assays that I'll show you that how we collect that information. But most importantly, for the interest of this talk, is we want to be able to look at these objects, these embryos, while they're alive in an individual well and extract any information we possibly can from the shape or the movement of the animal to say whether or not this chemical exposure that occurred early in development has hit something, modified a process to lead to an outcome. So the key is identifying these outcomes. So we and in terms to feed this workflow, we've invented lots of different ways to get unlimited amount of embryos to feed this system. We created ways to automatically, in mass, remove the outer eggshell to increase data flow. We invented robots that can automatically pick up embryos and put them in a multi-well plate. And we also automated direct chemical delivery so we could precisely put the chemicals across very tight concentration ranges in these multi-well plates. And we have another instrument we developed to capture this image. And that's at 120 hours. Right now, we use another commercial product to identify this behavioral responses in response to light dark stimulus or acoustic startle. So what you see here is we have a huge system, but anytime you do a system engineering approach like this, there's always a bottleneck. And as when you solve one of them, you create another bottleneck. And for us, and this is when we first met the Ramona team, is what was killing us is the imaging of these animals, and then secondly, to extract the morphology, the shapes of these animals in real time at a very high rate of speed. So basically what you're wanting to do, if you were an individual human looking through one lens, like Natalie said, you're going to look at this well and you want to extract this information from what you see. And prior, 15 years ago, we didn't do it with capturing with a microscope at all. We just used visual inspection with a low magnification stereoscope. So we had a group of people who were trained to see these features. And it's very, very slow. We didn't have a limb system. So, that was like the dark ages for us. So really the challenges were in this throughput is, we're measuring these things qualitatively with the trained eye of our researchers. It takes a lot of time to train and check the training of these individuals. And again, humans are, they have good days and bad days. So the inconsistency we were concerned about. And then interpretation bias. So you can have double checking of endpoints, but then of course you take a huge hit on your throughput. And then time is just our enemy. But again, we want to be able to look at these embryos at 5 days post-fertilization and tell whether or not they're anything different in them relative to the control. So what we did early on, and Natalie mentioned this, is first we did it by hand. And then we went to motorized inverted microscopes that we spent a fortune on. And honestly, our throughput killed the microscope. So not only were they way too slow that it was going to bottleneck us, even if we bought multiple microscopes, over time, having all these moving objects or moving parts in your imaging system, we were getting firmware errors. So that was a not a good solution. And then we thought we were clever. We went to flatbed scanners. So we took some high resolution flatbed scanners that you might see in an office store, and we kind of tore it apart. We were able to use these to capture reasonably good images from these individual wells by scanning the plates, but the quality was insufficient. So this is where Ramona comes on board. So we really wanted to do two things. First, we wanted to be able to capture the images without any moving objects very quickly. And so this is what we first implemented this. So we had a demo in our lab, and we purchased this the next day. It was that transformational for our workflow. So we've implemented this, and so not only did it improve our throughput of image acquisition, we were able to work with the Ramona team to take these images and basically cut them out and pop them into our limb system. So it became part of the metadata for our large database. So now the next hurdle that we are trying to tackle and are tackling with their team is really using machine learning techniques and using these high resolution images that we can now capture quickly and extract these features that I showed you in the previous slides. And so this is not trivial, right? It's pretty easy when you think about single cells in a dish that maybe are attached and adherent on the bottom, even more complex systems that don't move. And they're generally in the same Z plane, so they're on the bottom. Zebrafish, because they have swim bladders, they're moving. Some they move to different degrees. They end up in the water column at different levels. So because we can capture this entire plate so rapidly. We can anesthetize these guys. They generally go to the bottom, but they're not in the same plane. So we can with this instrument, it's feasible. It's not small data. We can capture a Z-stack across the entire plate, and we can segment the well. And then within the well, we can identify for each one of the objects which plane has the best focus to extract these images. So the key is you want to acquire the full Z-stack, the entire 96 animals at very rapidly. And we could do this in like a minute using the Kestrel. And then working with some data compression, you don't want to waste all the data on the information, the pixels on the outside of the image. You want to identify just the image and then feed this into a decision tree to identify the features. So this is not trivial to try to pull off moving objects, many of them individualized, but in a large plane, but we've been able to achieve it. So now working with these images and then working directly to identify first the object, the right plane where the object is, identifying these features, and then quantifying them using machine learning is really where we're at right now. So this is an example of one type of plate that we get, and you can see the type of analysis. So none of this software was available. So Ramona was able to work with our team. We provide access to our computer so we can do this type of analysis in real time. We can capture thousands and thousands and thousands of objects to make machine learning actually feasible. And this is kind of just showing the workflow and how fast it is. And then some of the things you might get images like this, and best case scenario, kind of expected scenarios. We're able to clearly identify this one is different than a control, but these are kind of odd shapes. But then these types of rapid capture provides the image examples that can feed the machine learning to further improve it in every iteration of our learning process. And we can identify novel features. So the speed at which we're advancing the segmentation with the Ramona team is really, I think, remarkable. So now another thing that we're working on, so it's a clever use of what we call these periscope plates. So again, these animals move. So you're being able to capture these animals as they're moving from the top and from the side. You can do this by putting a reflective surface. So you basically have one lens looking this way and another one looking down. So you can simultaneously see exactly where the animal is. This will absolutely overcome the orientation variability and challenge that we have in extracting these features. It definitely requires rapid capture, so we can grab these frames when they're in the right place to do the extract information capture. And in fact, what it does is it gives you kind of two shots at it, right? You can identify features from, say, the top plane and interpret that using machine learning. And then you could also do the same thing from the side plane. So you can make a consensus decision on whether or not, again, the original idea is, did the chemical perturb the development of this organism any way that we can capture the outcome? And again, this will absolutely increase the throughput and reduce for us the amount of labor we have in doing these manual assessments which I think is holding back, in my personal view, all of science. It's the cost of labor, which is reducing our discovery rate. So this, I think, is going to help us. This is something that we're moving forward in the future, is really being able to use this as a behavioral capture tool. And this is very challenging because of the data content and how big these files are. But being able to acquire behavior and morphological readouts in a single instrument could massively simplify our workflow and the workflow of others interested in this type of research. So this is really a fun use of the technology and allowing us to simplify the number of instruments we have to achieve the research goals that we have. And with that, I'll pass it over to Natalie and to Jason, and we'll take questions at the end. All right, thank you.
Awesome. Thank you, Robyn, for that amazing presentation. and for serving as an early client of Ramona's Kestrel system. I encourage everyone to put any questions in the Q&A function, and we'll answer as many as possible at the end of the session. Before we jump into Dr. Stein's talk, Jason, I'm going to take over for just a moment here to ask a quick pop quiz question. We'll give everyone a moment to answer here. All right, thanks everyone. Now I'm going to hand it over to Dr. Stein to describe how he utilizes Ramona's Vireo to image and analyze iPSC cortical organoids.
Okay, great. Hi, everyone. So I'm going to talk to you today about imaging cortical organoids to model interindividual variability in brain size. So our lab studies the development and structure of the human neocortex, which is the outer covering of the brain shown here. The human neocortex is greatly expanded in humans relative to other species. And it's thought that that expansion has allowed the development of human-gained cognitive and social abilities. And it's also thought that aberrations or alterations in the development of the cortex or the structure of the cortex can lead to different psychiatric disorders, for example, like autism. So our lab is interested in, what causes interindividual differences in the size of the human cortex. So one of the hypotheses about that, about what causes differences in the size of the human cortex is called the radial unit hypothesis. And this says that it's kind of down to early fetal development during the time period when the neurons are being born in the cortex. So this is an image of what the cortical wall looks like, a cartoon of what the cortical wall looks like during this period of fetal development from like 12 to 24 post-conception weeks when the cortex is being born. So during that time period, we have progenitor cells that exist, and those progenitor cells can make fate decisions. They can decide to form decide to have symmetric fate decisions to form two progenitors, or asymmetric fate decisions to form neurons. But neurons are postmitotic, they don't divide. So if progenitor cells decide to form other progenitor cells, which decide to form other progenitor cells, the overall progenitor pool increases, which increases the total neuronal production and leads to expanded cortical surface area. So this hypothesis was proposed to explain what caused human brains to have much bigger neocortices than non-human brains. But we think that this hypothesis is also relevant for determining interindividual variability within humans. So we're interested specifically in studying this early period of development when neurons are being born, and we use a human cortical organoid system for studying that time period of development. So the size of the cortex is also relevant for psychiatric disorders like autism, and here's an example of a study demonstrating that. So in this study, individuals were recruited who were derived from families who were at high likelihood for developing autism by virtue of having a sibling with autism, or that were low likelihood that did not have a sibling with autism. And the next child was followed with MRI scans early on in development from 6, 12, and 24 months of age, where pictures were taken of the brain to get measures of things like total surface area of the brain. So autism can be reliably diagnosed around 24 months of age. So here we're getting images of the brain prior to any kind of autism diagnosis. And what was found was that those children who went on to develop autism, shown here, had a cortical surface area hyperexpansion, so an increased size of their cortex early on in development that then led to an overall cortical surface area expansion later on in development. So this was an interesting finding, predictive of future autism diagnosis. So our goal is to try to understand the cellular and molecular basis underlying that cortical surface area hyperexpansion. So to try to assess these questions, we have been working on a project that we call the Infant Brain Imaging Study, the IBIS, Induced Pluripotent Stem Cell Study, iPSC. So this IBIS iPSC study, we recruited 18 participants who had previously undergone MRI imaging early on in development from 6 months, 12 months, and 24 months of age. From these individuals, then we got blood draws where we were able to get peripheral blood mononuclear cells, reprogram those peripheral blood mononuclear cells to induced pluripotent stem cells, iPSCs, which are cells that are like the embryo. So we're sort of going back in time to cells like the embryo, but that contain the same genetics as the individuals from which they were derived. Once we have those iPSCs, we can differentiate these iPSCs into cortical organoids, which are 3D systems that contain the cell types of the developing cortex during that time period when the radial unit hypothesis is kind of valid, during that early fetal development time period. So they're modeling that early fetal development time period. So using this system, we're able to uniquely assess the fidelity of cortical organoids for modeling inter-individual variability in cortical structure, because we have both an in vitro model of the brain and the in vivo brain growth of the brain as well. So during that time period, we acquired phase contrast imaging, 3D imaging, and single-cell RNA sequencing, though for the purposes of today's talk, I'm going to focus on this phase contrast imaging. So here's just an example of the 3D imaging to show you what the cortical organoids look like. The cortical organoids are a spherical shape, roughly spherical, and they contain progenitor cells, which are labeled by PAC6. They contain these lumens, which are labeled by N-cadherin, dividing cells labeled by Ki67, and topro labeling all cells. And here's just a 3D image where you can zoom around the organoid and then zoom in into each of these neuroepithelial buds, which are these neuroepithelial buds contain the cell types of that developing cortex, those progenitor cells that are making fate decisions to decide to form neurons. So this type of 3D imaging is very low throughput, but we wanted to also assess just a simplistic cross-sectional area of the organoids throughout the time of their differentiation. So we took phase contrast images of the cortical organoids at 0 days, 7 days, 14 days, and 35 days post-differentiation. And we segmented them as shown here. And then we, for each of the different 18 individuals that participated in the study, we get a trajectory of the growth of that organoid. From the trajectory of that growth, we can determine if repeated replicates of the same participant or different participants have more similar trajectories. And we find that the same participant has more similar trajectories than across participants, giving us some level of reproducibility of the findings. And interestingly, the growth rate of the organoid in a dish actually does have some correlation to the growth rate of the cortical surface area in vivo from the same individual. So these matchings between the in vitro measurements and the in vivo measurements, both growth rates and in this case the 24-month cortical surface area, so 2 years old cortical surface area, give us some indication that actually the size of the organoid does in some way correspond to the size of the brain from the individual from which it was derived. So this study was done in 18 participants, which was a lot of work, but still doesn't give a ton of statistical power to understand these relationships. So now we've been expanding our infant brain imaging study induced pluripotent stem cell project, the IBIS iPSC, to 115 different participants, some of which we have multiple clones per donor to assess reproducibility. So we made iPSCs from these already, where we sort of go back in time. And then now we're going forward in time to differentiation to generate these cortical organoids with the goal of trying to understand how in vitro measures of brain development correspond to the in vivo measures of brain development here. So of course, when expanding, we need a system that will allow us to image many different organoids, per individual participant, multiple replicates and across multiple different time periods. So our previous study used this system, which is the Thermo-Evo system. This is a microscope that fits in the hood, although it is totally manual. So you put the plate under the microscope. You can take a picture by pressing a button on the screen. You move the plate manually, take a picture, move the plate, take a picture. And this took us about 11 minutes per 96 well plate. Also, this system had no segmentation capabilities, so what we would do is take the images and then use cell profiler pipelines that we tried to develop, as well as ImageJ to do the segmentation. These worked, but they did have a lot of errors that needed to be manually corrected. So there was a lot of sort of back and forth to make sure that these were working. Now, with Ramona, we've been using their Vireo system, and we have much higher efficiency. So using the Vireo system, each 96 well plate can be imaged. You just put it in, the plate can be imaged in well less than a minute for a 96 well plate. And we get images of sufficient quality, of good quality for these. They can image both 96 well plates, and when the organoids grow up bigger into things like 24 well plates, they use also an inline segmentation system, so using a UNet and transformer that basically gives us much higher accuracy for segmentation. All of the segmentations still need to be manually checked, but they are much higher accuracy and require much less manual changes. So this is just an example of what our data looks like in high throughput. So with 87 participants, we've already differentiated and collected images on the Vireo system across multiple different days of differentiation, 1, 7, 14, and 18, and acquire the different cross-sectional images, cross-sectional areas from the images. And you can see that we have inter-individual variability here, which is kind of what we expect. We've also differentiated these out to 3 months, around 84 days, and we have 57 participants here, a few less participants just because this is an ongoing project and we have not yet completed the differentiations all the way out to the 3-month time period. And here you can see what the images look like, both for an organoid that's particularly large and one that's a little bit smaller. So our goal is to see if this inter-individual variability corresponds to the different sizes of the brain. We're currently blinded to those, so we don't actually have the relationship yet or the diagnostic status relationship, but that is our future goal, and we're able to expand to this size using the high throughput system. Another project that we're working on now is using these different cortical organoids, each of which have been genotyped across many different participants, and then exposing them to either environmental toxicants or clinically useful drugs, as well as vehicle, with the goal of assessing gene by environment interactions within a dish. So these gene by environment interactions can be well controlled and can tell us if genetic variants can modulate response to different environmental toxicants, which would be kind of a gene by environment interaction, or if genetic variants can modulate response to different clinically useful drugs, that would be kind of a pharmacogenomics approach. So these type of gene by environment interactions on organoid size can be done in really high throughput using this Vireo system, which we're excited to use for this. So here's just one example where we've used the system. So we've applied valproic acid, which is prenatal exposure to valproic acid leads to higher risk for autism and intellectual disability. And so we've applied valproic acid to our developing cortical organoids. Here you can see organoids exposed to either vehicle across different times, a low concentration of valproic acid or a higher concentration of valproic acid, and doing this across many different many different time periods here across multiple different organoids. We can see that there's a decrease in size across time, a dose-dependent decrease in size across time, which is consistent with what we know that valproic acid actually does lead to risk for microcephaly as well, to a smaller brain. Overall, this rapid parallelized imaging greatly reduced imaging acquisition time, which enabled us to scale our experiments to large numbers of participants and also multiple exposures. Also, the improved analysis enabled rapid segmentation, allowing assessment of organoid fidelity and response to exposures. And our future work using the system is to use fluorescent parallelized imaging to study neuronal activity in organoids via calcium indicators like GCamp across many participant lines and conditions. So just to give an example of that, this is not actually from our lab, but from Ramona, where they previously used GCamp imaging on organoids and shown that it is actually possible to image individual neuronal activity here. So we're very excited to use this in the future to scale up and to assess how different environmental toxicants or different genetic backgrounds can alter a neuronal activity in our model system. So thanks very much, and I'll stop sharing here. Happy to take questions too.
Thank you, Jason. That was a fantastic presentation. And thank you also for serving as our very first early access program participant for our Vireo system, which is now commercially available. So I'm going to give another quick pop quiz here for the audience on the speed of Ramona's system for Jason's workflow. Give everyone a moment here to answer this question. All right, thanks everybody. At this time, we're going to jump into some Q&A. So we'll start with one question from the audience. It's for Robyn.
Yeah, I see it.
Robyn?
Yeah, I see it, Natalie. So the question is from Samba. Samba wants to know if there's a possibility of using machine learning to assess the behavioral analysis. And certainly that's the case. We're working on that. We do have to remember, though, that this is a higher resolution instrument. So the types of behavior that you'll be able to acquire will be different than you might acquire from like a 15-minute video in some of the lower resolution. But absolutely, there are different types of behavioral endpoints that we are and will be able to apply machine learning tools to quantify those responses. It's a great question.
Awesome. Thank you, Robyn, for jumping in there. And thank you for the question, Dr. Parvez. So I wanted to ask a question that came up for both of you, Robyn and Jason. What was the learning curve for adopting this technology in your labs?
So I can take a shot at this. I think one of the really critical things about getting a new piece of technology that your students and technicians and postdocs use is whether they basically complain about it and how hard it is to use, or whether they sort of say like, wow, this has really made my life easier. And in this case, when the Ramona optics came in, it really reduced the amount of time that we had to do for the imaging part of this experiment. So there was really positive feedback on the imaging part. On the data analysis part, like that took a little bit more time, where we had to sort of work with the Ramona team to be able to get these accurate segmentations that didn't require a lot of editing, but they've been really responsive, and now we have like a really strong workflow that really requires very little very little manual interventions, although still does need to be manually checked for each of the images. So overall, it's been a really positive experience for us.
Yeah, I can.
Oh, sorry, Robyn, go ahead.
Yeah, I can echo Jason. I mean, I'm sure it's a very complicated system to build and to handle, the camera and lenses, but honestly, operationally, it's incredibly easy to use this instrument like Jason described in his talk. And I think what people don't recognize when you get this kind of this quantum jump in the ability to improve your workflow, it unleashes your imagination on other things you might be able to do. And I don't think people realize how much we limit ourselves based on how we've done things in the past. So I think what's fun with this type of technology is imagining, oh, now we can do this, and now we can do this. And since it's a simple system, it just becomes a lot of clever data scientists and image analysis people to really make the capabilities come through. And so I think it's been fun to feed our imagination is feeding their imagination, and we're building really cool stuff together. So. But that wouldn't happen if it wasn't easy to use. Sorry.
Well, thank you both for that feedback. We definitely try to make the systems as intuitive as possible. And Jason, you mentioned something about the human element in the segmentation workflow. And just to follow up on that, at Ramona, we talk a lot about using human-enabled AI and an AI-enabled analysis versus kind of pure artificial intelligence or machine learning. So can you talk about, where the human fits into that equation and to getting accurate segmentation data?
Sure, yeah. I mean, we feel like we should always quality check the data visually to be sure that we're getting sort of accurate results. So, the segmentation that's provided sort of you basically drag a little dot and then it sort of automatically segments the organoid around it. We're sure though to manually check those to be sure, and then you can do edits also to make sure if it's not accurate. So to either remove samples that are not accurate segmentation or to fix those samples. So we kind of like this sort of, you can look at it, you can see the data, you can see what it looks like and make sure that it's correct and then move forward. And that's the way that this system is kind of set up, which is really nice for us.
So question for both of you. How many different exposures are you able to test using this current setup? Maybe Robyn, you can start with this one.
Yeah, right now we do between 4,000 and 10,000 individual embryos a day. So it's pretty high throughput. And honestly, that limit is not hit by the image acquisition. It's hit by some other parts of our workflow. So I think we can even blow past that throughput in the short term.
We're considerably less throughput on that. So we have basically like around 115 donors, and we have 24 different environmental exposures that we're working on, or drugs. And so yeah, so but nevertheless, like that still results in a lot of plates for us. So this really has helped us to expand our workflow.
Great, thank you both. Question for Robyn. Are there certain alterations in the larvae that are missed? For example, those where orientation of the larva is important. I'm thinking of pectoral fins that are often best viewed in dorsal orientation versus mandibular deformities that are best viewed in lateral orientation.
Yeah, it's a great question. And this is, so some of you are probably aware there are other technologies where it's a slow technology where you can singulate an embryo in a capillary and kind of rotate it in front of a lens. That overcomes some of those challenges, but it takes such a huge hit in the throughput that that's not feasible for us. So I think there's two ways to look at this, right? So having the periscope plates are going to allow us to handle that problem, and we're going to be interrogating the animal in two different orientations in real time. So I think that'll solve that once it's fully implemented. The other thing is we've noticed from mining massive amounts of our data that the relatedness of endpoints to each other has led to us collapsing some of our endpoints that we don't need to collect them in order to identify an active molecule. So just statistically, we can reduce the need to see everything, but the periscope plate and the Kestrel allow us to get two different orientations at the same time.
Some follow-up questions here for both Robyn and Jason. Wondering if after you've used the systems, are there any additional features that you'd like to see in the future that might make your workflows even better?
I guess for me, I'm excited about the fluorescence in the future to use the GCamp imaging. I think that will be really, really interesting for us to be able to understand how genetic variability leads to differences in neuronal activation. So yeah, so excited for that. And then I think that will require some considerably more advanced segmentation tools across time, and if there's any movement in those as well, actually more similar to the stuff that Robyn's working on now. So yeah, I'm excited for that in the future.
Yeah, I would agree. We're Natalie knows this. We're working in the fluorescent space as well already. So I think that's going to be exciting when, as one of the advantages of ZBrush model is all the reporter gene lines that are available to us. Being able to do an entire plate of a 96 well plate or 44 well plate of embryos with different structures labeled and do that rapidly in automated fashion is really going to massively increase our different types of readouts that we're going to be able to add to our workflow in addition to morphology. So that's going to be great.
Awesome. So on our new Vireo system, we do have red, green, blue, and far red fluorescence options. And I saw there was another question about throughput. We can image all of those channels and bright field in 2 minutes for a 96 well plate. And all of that, timing can be optimized depending on your specific parameters, how many Z-stacks you're collecting, et cetera. And so we work with you to optimize that imaging and make it as fast as possible depending on your particular samples. And the Vireo also has environmental control. So we're really excited about these, GCamp or even, cytotoxicity type of live cell dynamics imaging that we can do at Vireo rate with continuous Vireo rate with continuous environmental control in the Vireo system. One question follow-up from Dr. Pervez, specifically for Robyn, but I think Jason can address it too. It's a really great question. You're getting so much data using the Kestrel system, most of which you don't end up using. What are some of the ways you are working on to extract the most relevant information?
Yeah, it's an evolution, right? So originally capturing the images was just feeding or reinforcing what the manual data collectors, the screeners that we call them, sitting at computers. But again, since we're capturing the Z series now, there is so much information, so many captures that are worthless, right? But being able to identify the worthwhile planes is really all we want. I think we're going to learn more when we start doing more of the behavioral machine learning to see whether or not I think we're going to have to keep more of that data, right? Because we're going to be capturing in different planes. I think for us, the key is to only keep what's useful. For data storage challenges, we have, petabytes of data, and yeah, quite expensive to manage that. So we're constantly trying to keep it manageable and useful so we can continue to do higher throughput assessments.
Yeah, I just echo that. Like we now had to pursue options for more storage because this does produce a lot of data. We have been, I think, a little bit less thoughtful than Robyn about, how to keep only the images that are important. And so, we've just been basically keeping everything that comes off of the scope so far. But, the downstream measurements that we're using are things like either overall cross-sectional area, is just one number, or things like circularity. I think reducing data down into like only the features that you need eventually will be something that will be important, especially if you scale up even more than like what our lab is doing. So.
All right, thank you both. Let's see, question for Jason here. You focused on describing organoid cross-sectional area as a phenotype, but are there other phenotypes you can derive from these microscopy images that can be used to assess the quality or fidelity of the organoids?
Yeah, so the cross-sectional area is basically just, understanding how big the organoid is. But sometimes organoids can differentiate, so have sort of off-target differentiations, and they can have like sort of cysts that grow off on the side, or they just basically are not as circular. So one sort of consistent measure of on-target differentiation is just circularity. So like if the organoid is more circular. So that's something that can be measured very easily using the existing pipelines that Ramona has developed. So that's great. Other features like that would be interesting to get in the future would be things like we can see buds, like these neuroepithelial buds, which I kind of showed in that 3D image. Those neuroepithelial buds are also indicators of on-target differentiation. They're basically forming that cortical wall that we're interested in. So it would be great to get kind of those pieces of information as well. And that would be something that, we do see visually, but we don't have like an automated quantification yet for those.
Okay, great. We've got a question here. If we want to conduct calcium imaging in swimming not fixed larval zebrafish in a 96 well plate or 24 well plate, which software system should we adopt? Perhaps I'll let Robyn speak to that and then I can chime in here.
Yeah, that would be really exciting to do. We've done that with confocal microscopy. And the Vireo system seems more equipped to handle that type of analysis. Yeah, we have not imagined being able to do that yet at this scale, but that would be really, really cool. I'll let you elaborate, Natalie.
Yeah, so we're obviously really excited about single cell capabilities of our Vireo system and moving to that higher resolution realm of 4x and 10x objective lens magnification. And so there are a lot of applications that we would like to apply that higher res system to, even for zebrafish outside of specifically cellular applications. And so we'd be really excited to collaborate with you on that and see if the resolution is sufficient for what you're looking for. We are already working with several groups who are interested in upgrading from the Kestrel to the Vireo. All right. Well, I think we've addressed most of our questions today. Thank you all so much for being such active participants. I do have one poll left that I want to leave you with here. So if everybody's ready to address this poll, just curious to hear about how you assess these systems for relevance for your applications now that you've learned a bit more about our technology and product options. All right. Thank you so much, everybody, for joining us today. And as we wrap up here, just a reminder that if you missed any part of the webinar or want to share it with a colleague, you will receive a link with the recording, so it'll be available on demand. We'll also follow up once we tally our poll responses and review our questions, and we'll reach out for those who will receive a prize today. And just really appreciate Jason and Robyn for joining us and sharing their firsthand experience on the system. And thank you to our audience for being such engaged participants. If you'd like to learn more about our systems, you can visit our website, ramonaoptics.com, where you can book a live demo or send us a message to inquire more about our technology. Thanks again and have a great rest of your day.